It's my enormous pleasure to welcome everyone here. This is our 8th annual research symposium. And for me, I was getting very excited about this meeting because I always learn so much. Sorry, I have to remember to stay here. You know, it's very hard to keep up with the breadth of what Archbold does. And this is one of the rare occasions where one sees all of these tendrils of great facts and new information coming in. So I hope you share in my excitement about learning today. I'm going to take a couple of minutes to welcome everyone here and really making a big recognition that we're a regionally dispersed group. Thank you for everyone coming up from the ranch. Thank you for everyone coming down from Avonpark Air Force Range. We really appreciate the commitment to coming here for the day. I'd like to invite, I know we have some local visitors who are not part of our staff with joining us today. Danielle, I saw you walking in today. Thank you for joining us. Others that I may have missed. It's really wonderful to see folks like Eric coming back, Eric Menges from emeritus plant ecologist here. It's really great to have you here, and he's going to be around next week, so thank you. Chris and Abe, what year is this, Chris, for returning to Archbold? 52. None of you are going to outcompete that. But lovely to have you here, and I'm so glad that your time overlaps with this symposium and still all actively contributing to what we're doing. Also, a big shout out to the chairman of our board. Leila, could you stand up for a minute? Leila Love, yay! Thank you for taking the time and expressing the interest and joining us for a symposium. It's wonderful to have a board and a chair who is really interested and, will stick it out through a symposium to learn what we're doing. So thank you for being here. Okay, so Laura, I can't actually see who's online, so maybe you could talk me through numbers and who's there. So first of all, welcome to all 19 online, the Archbold diaspora. Thank you so much for being here. So both to the two Johns, John Fitzpatrick and John Thaxton, thank you for being here. I know you'll share my enthusiasm. I know John Fitzpatrick was planning to be here, and he's been waylaid by some peripatetic scrub jays today who are moving from Ocala to Jonathan Dickinson's State Park. So look after them really well, please. All right. Also my job to just roughly go over the day. There is a PDF in your calendar appointment, or Laura can give you information, or I have a hard copy which we'll share. And you can see this sort of real cross section. I'd like to give a huge thank you to the people who helped organize that, and that is Joe Gentili and Laura. Thank you both very much for doing that. It's never trivial to wrestle all of those emails into a cohesive schedule for the day, so we really appreciate it. And Laura for all the online side, both of you, thank you very much. So we're going to start out, we should be kicking off the first talk at 9:10 with, his first talk is by Josh Daskin. We're not going to introduce everybody, so as you come up, introduce yourself and your talk, and be standing by to be the next ready. I think Laura's got everything preloaded, yes? Okay. Okay, so we'll need a minute or so to change those over. All right, so give yourself a bit of time to change those over, and we'll just do the self introduction, so it'll save a bit of time. So thank you for that. We're going to have a break from 10:15 to 10:30. Although I'm not introducing you, I will be a fierce timekeeper for you. So, and those who know me know I am a fierce timekeeper, so be warned. Each talk is scheduled, as shown, and as we all know, it's hard to give a 15-minute talk. Leave a couple we're going to do questions as part of the talk as well, so maybe about 12 minutes with 2 to 3 minutes for questions. Yeah. Yes. So Laura will be, if there are questions and time at the end of everybody's talk, Laura will be forwarding those questions via me to answer. Is that correct? Yeah. Okay, so break from 10:15 to 10:30. We're going to have a noon lunch break, and for those of you who would like to eat pizza, there'll be a surfeit of pizza, so be ready to join in that. Then from 1:30 to 3, a break at 3 o'clock, and finish at the end of the day. Betsy Boughton, I think you're bringing us home. Always worth staying for Betsy. So, okay, so I'm going to go ahead and get the first speaker up, who is Josh Daskin. And the person after that is going to be Jenny Schafer, who I presume is online. Is that right, Laura? Okay. So Josh, over to you and self-introduction.
All right. Good morning. I am the...
One thing you have to do, you have to share your screen now.
Okay.
To Zoom.
While we get shared to Zoom, I'm Josh Daskin. I'm the Director of Conservation here. And because I have the pleasure to open today, I'm going to try to give kind of a lighter talk, a little more fun. It will be participatory. So hopefully everyone has come and is wide awake. You may need like a phone or a piece of paper to keep track of some things as you go, but you'll get fair warning for that. So all of you have also contributed to today's talk. I'm going to be talking about our budding effort to track our conservation impacts. And so this is part of our conservation strategy that has been developed collaboratively across all our programs, our labs, our education program, and with our board members as well. So this is sort of an update. And some of you in the room have been involved in this since the very start of it 2.5 years ago. Some of you are new to Archbold. And so I'm going to give a little bit of introduction to what this is, and then we'll dive into some of what we're learning. So as a reminder, we have our 4 conservation pillars, and these are kinds of the things that the buckets into which we place a lot of our work, and especially our more applied work: saving the rarest of the rare species and ecosystems, sustaining both working and natural grasslands, addressing climate change, and connecting large landscapes and wildlife corridors. And we want to use our science and our education efforts to move the needle on all of these 4 pillars. We've always done this. We've always been working on these topics, but somewhat more reactively and not as maybe as a synthetic whole as we are now. And so we're trying to move towards measuring these impacts organization-wide. And that is tricky for a couple of reasons. Firstly, we have a very diverse organization working in multiple places on lots of different topics, including, for instance, species conservation, ecosystem services, agriculture, education. We have long-term responses that are difficult to measure. So Betsy Rothermel is tracking tortoises that live 100 years. How do you measure our impact on species so long-lived? We're doing, put the microphone on. Thank you. We are doing long-term climate change resilience planning in the Avon Park Air Force Range and the Sentinel landscape around it. That takes decades. Other folks here are working really at the grassroots level. So Dustin's whole program is about teaching young folks to care about conservation, and that may also take decades. We have indirect impacts. So I'll be driving up to Tallahassee this afternoon to talk with legislators about conservation and conservation funding. How do we measure one organization's impact when there are 15 or 30 groups that are working towards some of these things? And so these challenges are not unique to Archbold. Many groups, a few of them represented here, including LTAR, which we're involved in, for-profit companies, and academics who've written books about this, are working on how do you measure conservation impact. There are some tools which many of you have participated in using. So here's one example of the never pretty to look at, but always useful strategy pathways, which the plant lab developed with me. This one in particular for looking at how they're influencing Florida ziziphus, a really rare plant, conservation and restoration. And these purple boxes, those of you who, the purple triangles, those of you who have contributed may remember, those are where we're kind of inserting the metrics that we want to measure for each of our pathways. We're using tools called the conservation standards, and they come from a book called Pathways to Success. So if anybody wants to dive into details, I'm happy to talk with you about them. So at this point, all of our programs have developed a series of metrics, things that they want to measure going forwards that are tied to one or more projects. And we'll be using these to understand some, not all, because you can't measure everything at all times, some of our conservation impacts across our 4 pillars. I have bucketed these metrics into 3 types of impact. So the gray box, building the evidence base, that's our science. How are we measuring things? Outreach and capacity building, how are we training folks? How are we educating people about conservation? And conservation outcomes, that's like, how are species doing? How are ecosystems doing? What's actually happening on the ground for the conservation values that we care about? So now comes the participatory part. Because it is football season, and everyone should be getting ready to bet on the big game, you will have a chance to wager on a series of, I think it's 14 of our metrics. So we have first for the building the evidence base, and you may want to have something to keep track of where you're at. I have 1, 2, 3, 4, 5 for evidence base here, and you have to tell us whether we had over or under each of these numbers. So I'll give folks about 60 seconds to review this one, and then we have 1 slide for each of the other metric types. Don't take this too seriously. There is no prize for the winner. Most of them are annual time frame, yes. 2023. Feel free to discuss with your neighbor if you feel you need help. Give folks about another 10 or 15 seconds on this one. Okay, doing a little Zoom repair in the meantime.
Okay, that should be better.
How about now? Can people hear better online? All right, last 2 seconds on building the evidence base. So tally up how you did. Particular shout out to the ranch, which had the highest single number in any of their metrics for almost 570 million. All right. Outreach and capacity building. We have 3 metrics here for you to work on. The number of folks trained as firefighters, total education participants that Dustin and his crew have reached this year, and the number of events we have hosted or participated in forwarding the vision of the Florida Wildlife Corridor. Right, 10 more seconds just because we only have 3 of these. How'd you do? Dustin's reaching 12 people a day, every day. And now conservation outcomes. So these are all the kinds of values that we are that we're tracking, ranging from climate change at the station to scrub jay populations here at Archbold, and a bunch of offsite locations that folks are monitoring with Sahas. About another 20 seconds. All right, and here come the outcomes. So the jays are trending upwards in the places that we monitor. We're getting close to 60,000 acres contiguous conserved area. All right, so folks should now have a tally of how many they got right. We had 14 total. Did we have anybody with all 14? Anybody 10 or better? We have a winner. All right. We'll see if anybody can beat Hilary next year. So why are we tracking these metrics and about 55 others? We have about 65 total. Having these on hand going forwards, especially as we start to build longer timeframes and see trends, will help us understand the status of our conservation values, both here at Archbold and in our conservation neighborhood. I know Talia is very interested to use these to sort of increase the visibility of our programs, which helps with both public and private funding support. Many of these funders are interested not only in the critical basic science we do, but also the impact we have. And so next steps for the conservation strategy and our metrics are to, number one, update the actual strategy itself, which is available on our website for folks who want to see the details, to set goals for our flagship metrics. So we're going to take about a dozen of the 65 that you all have submitted and say, these are ones that are indicative of broader trends. So for instance, species that require really good habitat in the scrub, frequently burned according to a planned schedule at least. Their trends upwards may be indicative that there's lots of good scrub habitat out there. And scalable, we want to be able to understand trends not only on our 20,000 acres, but perhaps across the 1 million acres almost of the Avon Park Sentinel landscape, or the 2+ million acres in the Everglades headwaters. So we're going to look for metrics that we may be measured at Archbold scale or Archbold in the neighborhood scale this year, and then eventually be thinking about how do we track the same things over a larger landscape. And then we'll be working across programs and with our communications folks to message our impact to partners going forwards. So thank you all for coming first thing in the morning and for being willing to play my game and hopefully get your brain moving for some deeper science the rest of the day. And I will leave it there. You want me to leave it screen sharing?
Actually, no, because we have.
Hi, Jane.
Hello.
Okay, that did not work from my computer. So we're ready for you to share your screen.
Okay. I don't see the regular share screen option that I normally have. So how should I go about doing that?
Well, and we go back to my computer. Oh, it didn't make you a panelist for some reason. Okay, hold on. It let you talk, but didn't make you a panelist. Give me a second.
Okay.
Okay, should work now.
All right.
And I didn't think about this part where I have to share the correct screen for the people here to see.
Okay, I think I'm sharing my presentation, at least from my view.
I don't know. The easiest way to do that is like this. So I apologize for the people who are here. I will figure this out. That break. How's the best way to fix this? Okay, everyone should be seeing you now.
All right, so I'm good to go?
Yep.
Okay, great. Thank you. I'm Jenny Schafer, and I am an associate professor of biology at Winthrop University, but I have been coming to Archbold since 2001 when I was an intern in the education program, and then I was an intern in the plant ecology program as well. So shrubs can create patches of high resource availability within a landscape, and these patches are called fertility islands. The availability of nitrogen, phosphorus, and other nutrients is often higher under the shrub canopy than in open areas among shrubs due to the funneling of organic matter and precipitation under shrubs. The fertility island effect has been well documented in arid and semi-arid ecosystems. This picture here is showing creosote bushes in the Mojave Desert. The abundance of annual plants under the shrubs is greater than their abundance in the open areas among the shrubs, in part due to high resource availability under the shrubs. After I conducted my research, my dissertation research at Archbold, I conducted some research in the Mojave and Sonoran deserts as a postdoc, and we focused on the fertility island effect. That was over a decade ago, and since then I have been interested in investigating the fertility island effect in Florida scrub, and I was finally able to do that this past summer working with 2 undergraduate students from Winthrop. Florida rosemary scrub is dominated by Florida rosemary, Serratiola ericoides. There are open gaps among the shrubs, and this is a characteristic of rosemary scrub. There is greater open area among shrubs in desert ecosystems than in rosemary scrub, but the growth form of rosemary is somewhat similar to that of creosote bush. There are patches of shrubs within the landscape, and litter does tend to be concentrated under shrubs. So we tested the hypothesis that shrubs in rosemary scrub create fertility islands. We focused on 2 questions to address this hypothesis. Fire is not a historical disturbance in desert ecosystems, but fire is necessary to maintain populations of the endangered endemic herbaceous species in Florida rosemary scrub. So we were interested in if the ability of rosemary to create fertility islands varies with time since fire. And we also investigated if shrub species differ in their ability to create fertility islands. So first, I'm going to talk about if the ability of rosemary to create fertility islands varies with time since fire. Rosemary recruits from seed after fire, and so we were interested in how long it might take for rosemary individuals to grow large enough to create fertility islands. To answer this question, we worked in 3 rosemary scrub sites. One burned 8 years previously, one 13 years since fire, and one 28 years since fire. This graph illustrates our prediction. We predicted that rosemary shrubs would create fertility islands and that their ability to do that would increase with time since fire. So we expected to find a significant interaction between location, open versus rosemary, and years since fire. And we expected the difference in nutrient availability between rosemary and in the open to be greatest in the 28 years since fire sites. So we haphazardly selected 8 rosemary individuals in each site. We measured shrub size and litter depth under the shrubs. As you might expect, shrub canopy area, height, and volume all increased with time since fire, as did litter depth under the shrubs. To measure nutrient availability, we used cation and anion plant root simulator probes. In the 13 years since fire site and 28 years since fire site, we established 20 by 20 centimeter plots under the shrub canopy and in open areas among the shrubs, also 8 areas adjacent to the focal shrubs. In the 8 years since fire site, we established plots in the open areas, but there was not enough space under the shrub canopy to fit plots. In that site, we put the probes just around the base of the shrubs. In the other sites, we buried the probes in the plots. And they were in the field from May through July 2023 for about 2 months. All the graphs for this question will be oriented in the same way. Years since fire is on the x-axis, nutrient availability is on the y-axis. The units are micrograms per 10 centimeters squared per 65 days. So that's the amount of the nutrient per the surface area of the ion exchange resin per the length of time the probes were in the field. The factors in the analysis are under the legend, and a star next to any of those factors indicates a significant effect. The bars indicate the mean, and the air bars are the 95% confidence intervals. So this graph is showing total nitrogen, which is the sum of nitrate and ammonium. The majority of nitrogen was in the form of nitrate. So the nitrate patterns are similar to the total nitrogen patterns shown here. We found a significant effect of location and years since fire. Overall, nitrogen availability was higher in the open areas among the shrubs than under rosemary, and nitrogen availability decreased with time since fire. This graph is showing phosphorus availability. We found a significant effect of the interaction between location and fire. So phosphorus availability was higher in the open areas than under shrubs in the 8 years since fire site. But in 28 years since fire site, phosphorus availability was higher under rosemary than in the open areas among the shrubs. This graph is showing potassium. There was a significant effect of time since fire and a significant interaction. Overall, potassium availability increased with time since fire. Potassium availability was higher under rosemary than in the open, but only in the intermediate and longer unburned sites. And this graph is showing calcium availability. The patterns for calcium availability are similar to those for potassium. There was a significant effect of fire and a significant interaction. Calcium availability was higher under rosemary shrubs than in the open in the 13-year and 28-year since fire sites. So does the ability of rosemary to create fertility islands vary with time since fire? Overall, I would say yes, but it does depend on the nutrient you're focused on. So rosemary did not increase availability of nitrogen under its canopy. In fact, nitrogen was higher in the open areas among shrubs than under rosemary. There are a couple of potential mechanisms to explain this pattern. The biomass of rosemary roots is highest under the shrub canopy and surface soils. And so high uptake of nitrogen by the roots could decrease nitrogen availability under the shrub canopy. In addition, rosemary is allelopathic. It has negative effects on germination and growth of the endemic herbaceous species. Transformations of nitrogen in the soil are driven by bacteria, whereas transformations of other nutrients such as phosphorus are more influenced by abiotic factors such as soil characteristics. So it is possible that rosemary could have allelopathic effects on the bacteria in the soil underneath its canopy. For phosphorus, potassium, and calcium, rosemary did vary in its ability to create a fertility island with time since fire. Rosemary increased availability of phosphorus, potassium, and calcium under its canopy, but only in the 13 or 28 years since fire sites. And this is likely related to the shrubs being larger and being able to funnel more moisture and more organic matter under the canopies. So for the question, do shrub species differ in their ability to create fertility islands? We focused on rosemary, Lionia fruticosa, and Sabuletonia to have 3 species that differ in their growth forms. To address this question, we worked in the 28 years since fire rosemary scrub site. This graph illustrates our prediction. We expected all shrub species to create fertility islands, but we expected rosemary to have the biggest effect on nutrient availability and increase that the most under its canopy because it has the most similar growth form to creosote bushes, which are known to create fertility islands. So we used the same rosemary shrubs that I have talked about previously in the 20 years since fire site, and we also haphazardly selected 8 Lionia and palmetto individuals near the rosemaries. We measured shrub size. Rosemary and Lionia have larger canopy areas than palmettos. Rosemaries are intermediate in height compared to the other species, and overall, rosemary and Lionia have a larger shrub volume than palmetto. We used the plots we established in the open and under rosemary, and we established plots under Lionia and palmetto as well to assess litter cover and depth. Overall, litter cover was higher under rosemary and Lionia than in the open, and palmetto had intermediate litter cover, but litter depth was higher under all shrubs than in the open areas among the shrubs. We measured nutrient availability using the cation and anion plant root simulator probes. These were buried in the soil within the plots under the shrubs or in the open, and they were in the field for approximately 2 months. For this question, all graphs will be oriented the same. Again, the nutrient availability is on the y-axis in the units of micrograms of nutrient per surface area of the exchange resin per length in the field. The location is on the x-axis. The bars are the mean, the 95%, the error bars, the 95% confidence intervals, and the star indicates nutrient availability that is different from the open. So this graph is showing total nitrogen, which is again the sum of ammonium and nitrate. And we found that nitrogen availability was lower under rosemary and under Lionia compared to open areas among shrubs. This graph is showing phosphorus availability, and phosphorus availability was higher under rosemary than in the open, but phosphorus availability under Lionia and palmetto did not differ from the open areas among shrubs. This graph is showing potassium availability. Potassium availability was higher under all shrubs than in the open areas among the shrubs. And for calcium availability, it was higher under rosemary and Lionia than in the open areas, but there was no difference in calcium availability under palmettos compared to the open areas among shrubs. So do shrub species differ in their ability to create fertility islands? Yes, they do. Rosemary increased the availability of 3 nutrients under its canopy, while scrub palmetto only increased the availability of 1 nutrient under its canopy, though there was a non-significant trend towards higher nitrogen availability under the scrub palmetto canopies. Differences among species are likely due to differences in their growth form and nutrient uptake under their canopies. Rosemary has more branches, which could potentially funnel more precipitation under its canopy, tends to have a more compact growth form, which concentrates litter under the canopy. In contrast, palmetto has kind of a broader growth form with leaves fanning out from a central point. Precipitation can drip off the leaf tips, kind of outside the canopy, rather than necessarily moving down the petiole towards the center of the stem. And when the leaves die, they fan out from the plant and can often then lead to litter not accumulating as much under the shrub. Also, species could differ in their uptake of nutrients under the canopy. Rosemary and Lyonia have associations with ericoid mycorrhizae, and those mycorrhizae could be particularly efficient at acquiring nitrogen from the soil, which could contribute to lower nitrogen under the canopies of those species. So overall, our hypothesis that shrubs and rosemary scrub create fertility islands was supported. This could have important consequences for community and ecosystem dynamics in rosemary scrub. Patches of high resource availability could provide microsites that are favorable for growth of other scrub species. In the 20 years since fire site, we found a higher number of understory species under palmettos than the other species, but we did find some species growing under rosemary shrubs. So more research is needed to tease apart the negative impacts of the allelopathy of rosemary and the positive impacts of high nutrient availability under shrubs.
Jenny, I'm going to ask you to wrap up now. Oh,
Oh,
perfect.
Just finishing, yes. So I just like to thank Aaron David and the Plant Ecology Program for hosting us and providing logistical support and our funding source through Winthrop. And if there's time, which maybe not, I could take a question.
Maybe we could take one question and then. Okay, we'll move on. Thank you very much, Jenny.
Thank you.
Bear with us while the Zoom queen is sorting things out.
Let's go people. Try a bit.
Pretty nice on.
So you can go ahead and start introducing yourself.
Okay. Hi. We'll wait for you. 2 minutes. Okay. Good morning. I'm Karen Rice David. I'm a research assistant at the ranch in the agroecology lab with Betsy Boughton. And today I'm going to give you an overview of a project we've been working on for the past year with David Royal from the Nature Conservancy, looking at overseeding for soil health and soil water quality.
Okay.
Does this work?
Yeah.
Okay. All good? Good. Okay. So of the 2.6 million acres in the Everglades watershed, 1 million of those acres is ranchlands, is comprised of ranchlands. That makes ranchlands very important to the nutrient runoff from ranchlands that get into these really sensitive ecosystems. And a major way that ranchlands contribute to this nutrient runoff into the watershed is through legacy phosphorus. Legacy phosphorus is phosphorus that was used as fertilizer decades ago. We no longer fertilize with phosphorus at the ranch, but it continues to persist in the soil. And if plants can't access that phosphorus, it can leach away into the waterway. When it gets into the waterway like Lake Okeechobee, we can start seeing algal blooms, which is really detrimental to the ecosystem and the health of wildlife and humans. So we need new ways to capture that phosphorus, to get it out of the soil, prevent it from getting into the water system. And one idea is to use overseeding. And overseeding is when you plant something directly into existing pasture land. So here is our ranch, not the greatest picture on the screen, which is all bahia grass, but this is rows of sun hemp that's overseeded into the bahia grass. So that pasture remains as it is, but new species are popping up. And if we can overseed a diverse mix of species into our pastures, we might be able to have different benefits to capture those nutrients. Things like legumes can contribute nitrogen, deeper root systems can scavenge nutrients that are deeper in the soil that the bahia grass can't access. Big taproots like radish and turnips create holes in the soil to allow more aeration and water flow. When we plant cool season and warm season crops, we can maintain that active growing season year round. And overseeding has been studied a lot in the Midwest and even northern Florida. and it's shown to really maintain high forage quality for the cows on the pasture. But we don't really know if it can capture those nutrients, and we don't know if they can actually grow well in this part of Florida because we have such an extreme climate. So this is a 3-year project, and we want to answer 3 questions. Can these overseeding of diverse seed mixes actually be established in this part of Florida? If we can establish them, can they improve soil health? Do things like improve the nitrogen availability, increase the organic matter and the active carbon? And can those diverse seed mixes improve water quality, specifically by having that plant community uptake that legacy phosphorus in the soil? So this is Buck Island Ranch, and in green, those are our 40-acre control pastures. They receive no treatment. We also have 3 40-acre treatment pastures, and each treatment pasture is divided into 3 13-acre sections, and they receive a unique seed treatment. And these seed treatments were developed by the Nature Conservancy and the Seed Producer Seedway. They created these mixes, determining what would survive the best in this area and accomplish our goals. And these are all in improved pastures. That means that it was planted with Bahia grass. That's the dominant grass that grows there. So our seed mixes include Winter Mix 1. This was first planted in November 2022. You can see the functional diversity that we're going for in this mix. We have crimson clover, which is a legume, the large roots of the radish and the turnip, and we have rye with its fibrous root system. We have another winter mix planted at the same time as Winter Mix 1, but there's 7 species. Again, we have some legumes. We have crimson clover and hairy vetch. We have the daikon radish and the turnips, and we have 3 grains, triticale, wheat, and oats. And then our third mix is a summer mix. It was first planted in April of 2023, and we have 3 species here. Ashenominy, which is actually a native legume. That's our partner, David Royal. He's holding pearl millet, which is a bunch grass, a lot like sorghum, and he's holding sun hemp, which is a deep-rooted legume. We collect data on species composition. We look at the percent cover of all species growing in our plots. We look at forage quality, and we collect primary productivity data. We also collect root biomass. We collect root biomass from 4 different depths, going from 0 to 60 centimeters in depth. Not only can we look at the total root biomass, but we use WinRhizo software to scan the roots we collect. We can look at the proportion of fine roots to coarse roots. Hopefully, if we see more fine roots, those are the roots that are going to capture those nutrients for us. We also use the same probes that Jenny Schafer just told us about to look at nutrient flow in the soil, and we look at soil moisture. We collect soil from the same 4 depths like we do with the roots, down to 60 centimeters. Some of that soil we send off to Cornell. They give us a soil health assessment. So for different soil health indicators, they give us a value of that indicator, things like active carbon and aggregate stability, and then they rate that value for us to tell us what's optimal for plant growth, what needs to be improved, what might be constraining plant growth growing in the pasture. We also send soil away for phosphorus analysis. So we've had 1 year of this project so far. Each of those mixes were planted once. Do we see if they can be established? Well, in our randomly selected plots within our pasture, these are 1 meter diameter plots. We record if these species that were planted are present or absent. And you can see these tables show us the different mixes and the percent of the plots where the species was actually observed. If it wasn't observed, that means either it didn't germinate, or if it did germinate, it died before it could be included in our census. Things were not looking great for year 1. We have on average, we have out of 11 species, 4 species are observed more than 70% of the time. And another way to look at this is the percent cover contributed by those seed mixes. So in our 1 meter diameter plots, for the percent cover on average, was mostly Bahia grass, to be expected. Smut grass contributed a lot to our percent cover in our plots. That's an invasive bunch grass that we're trying to control at the ranch. And in yellow, you see where our different seed mixes all contributed. So on average, it was 2 to 4% cover. It's not a whole lot of percent cover. So for year 1, we didn't see great establishment of these. We think that Bahia grass might just be too competitive. Here's a picture of our recently planted winter mix. And you see on your left, that's nice rows of planted winter mix coming up. That's where we drive our trucks a lot to get to the pasture. As you get into the actual pasture itself, with a lot of Bahia grass and smut grass, you really visually just don't see it growing anymore. And then we have in red on the map, those are 72 acres that are extra acres where we planted the summer mix and the winter mix. And anecdotally, when we saw things growing there, we had radishes bigger than my hand. In our improved pastures, our radishes were so tiny. So there's something about that really disturbed soil that is helping things thrive, and they're just not able to do that in our improved pastures, at least for year one. So for year two, we have prepared a little bit more. We grazed really heavily before planting. So we tried to get that Bahia grass competition lowered. We also had really low pH in year one. We had an average pH of about 4.5, which is actually too acidic. So we added lime. That's the truck that adds the lime. And so we're hoping to raise that pH to help those species thrive a little bit more. We also have different weather conditions. After year one, or in year one, we had aerated the pastures, like all the soil. Tropical storm came, and a month later we got to plant. So this year the weather was better. We were able to graze, aerate, and plant. So we're hoping things are better for year two. When it comes to if these mixes can improve soil health and water quality, we aren't really expecting a lot of changes because the seed mixes didn't establish, and we don't have that data yet. But I will show what we saw before planting occurred. So this is data from our Cornell soil health assessment. In green are soil health indicators that are optimal for plant growth. In orange and red means those really need to be improved or they're really constraining plant. And we saw that 65% of our total root biomass is in that top soil layer. So if you're a plant, you're pretty happy at the ranch. The soil's moderately healthy because that's where your roots all are for Bahia grass mostly. We really could use improvement in those lower soil layers. Hopefully diverse seed mixes can help improve the soil down below. From a soil water quality perspective, our deepest soil layers seem to be the most problematic. So this figure is showing you the average water extractable phosphorus, and that's phosphorus that's labile in the soil. It's readily subjected to leaching when there's flooding. And you can see that our lowest soil layer, that 45 to 60 centimeter layer, has the most average water extracted phosphorus. And this is unfortunate because that's the least amount of roots in that layer too. So there's really nothing there to capture this phosphorus that's just able to leach whenever we have too much water in the soil. Another way to look at this is looking at average soil phosphorus storage capacity. This is how well our soils are able to actually hold on to phosphorus. The dashed line is where 0 is. Above 0 means we have an increasing potential to store phosphorus. That's what we want. Below 0 would be an increasing potential to leach phosphorus. And this is an average of our pastures before seeding, just averaged all together. And averaged together, they look okay at storing phosphorus. They're mostly above 0. There's no significant difference between the soil depths. However, when we look at them by block, we see a different story. The top figure is our northern block, and that's a Bassinger fine sand. We saw no significant difference between the soil depths, and overall it's able to mostly store the phosphorus. Our central block is an Immokalee sand, and we see that there is a significant difference in the soil depths. That lowest soil depth has the potential to leach phosphorus. And as you get closer to the soil surface, you have a small potential to store phosphorus. Our southern block is mostly a Tequesta muck, very organic, very wet pasture. And there we see that that top soil layer with a lot of organic matter has a strong ability to store phosphorus. But as you get lower into those soil layers, we start leading toward the ability to store less phosphorus, possibly even leach the phosphorus. For conclusions, can we successfully establish diverse seed mixes that we overseed in the pastures? We didn't see great establishment in year one. However, we took steps to try to improve those establishment. There are species that did seem to germinate well, at least. And some of these, especially something like ash anemone that occurs natively here, might be able to seed itself back in, which would also help a rancher out, that they don't have to seed these in every year. They can just start growing themselves. And we're also looking at ways to explore using overseeding in something like after a burn or after there's another disturbance on the ranch to try to kind of get around that competitiveness from the Bahia grass. We saw that our surface soil layer was moderately healthy for plant growth, but we could really see improvements in soil health deeper than that. And that deeper soil layer really poses a threat to soil water quality. And we see a lot of variation throughout our pastures, throughout the ranch in the different pastures. And the ability to store or leach phosphorus is probably soil type dependent. So we have just finished planting our second round of our winter mixes. We're starting to see them grow now. These are pictures taken a few weeks ago. We see a lot of radish and turnip coming up. So we're going to survey those during the end of this week. And we are working with an economist to see if the cost of actually overseeding these species outweighs the benefits. So we're working on that right now. We're also starting a shade house experiment trying to relate soil moisture to germination, just to try to pinpoint the best soil moisture for planting that might help these species thrive. And we're looking more closely at the different species themselves and the mixtures to see how nutrient flow is impacted by the different species. This is funded by FDACS and the National Cattlemen's Beef Association. And I just like to thank the research staff and operations staff at Island Ranch. So many people help collect this data, and operations is the ones who actually are out there aerating and planting and grazing. And we get a lot of help from GIS department and others at Archbold Biological Station. Yay! I don't want to touch anything.
Really good timing, and maybe we've got time for a couple of questions. Go ahead. You go ahead.
Josh.
I was thinking, get a sense of how percent coverage you actually get these established, but 2-4%, anything like that.
I know. Yeah, the question of can we establish these, it's kind of arbitrary to say, well, 70% of the time when we see it, that's good, or 4% cover isn't good. Yeah, that's hard to say. I guess from our perspective, you know, when you go out there after you see all that planting, you see how nice it is. Like, it's literally, all those pictures showed you, like, it's these rows growing up, and then you go in the pasture and you don't see much. It's hard to say what really is a number of establishment, but with the cost, I think that's what the economist can help us with. Because if you're buying seed and then you only see it every so often throughout the pasture, is it going to be worth it for a rancher to overseed and buy that seed? So the rancher has to pay that cost. Also with our ranch, even within pastures, we see a lot of heterogeneity. And so I think you'd want as much to grow up as possible because you're not sure where in your pasture you're going to have those like hot spots of phosphorus. So yeah, that is a question of like, what is successfully established?
Maybe time for one very
So that's improving the diversity of plants in the pasture, improve the water quality from the cows off.
Yeah, so Bahia grass goes somewhat dormant in the wintertime. And so then those cows are moved off into the semi-native pastures to graze. So having all of these species planted have much higher like crude protein and digestibility than Bahia grass alone. So that is better for the cattle. Hopefully they can kind of stay in the same pasture and graze those. So yeah, it helps. That's a lot of what overseeding is used for, is to help maintain forage quality.